Good afternoon. So we should start. I know that there are still people joining this webinar. So this is the 5th episode of our ESHG webinar series. I'm Bill Newman. I'm the immediate past president of the ESHG. And we set up this webinar series earlier this year to showcase some of the wonderful work that's going on in genomics across the world, predominantly in Europe, and to give some of our esteemed colleagues, both within the Scientific Programme Committee and out with an opportunity to present on different topics that we think would be of interest to our membership. And so it's my absolute pleasure today to introduce our 5th speaker. It's my friend and colleague, Sid Banka. So Sid trained in medicine in India and then moved to the UK and undertook a number of jobs before ending up in Manchester and taking up a career in clinical genetics, and has undertaken a PhD and then established his own clinically orientated research group in developmental disorders, and along the way has taken on a number of senior roles. So he's the clinical director of the Manchester Centre for Rare Conditions. He's one of the co-leads for the NHS England Network of Excellence in Rare Diseases. And he's made a big commitment to the ESHG, so sits on the Scientific Programme Committee, but also leads the ESHG course in dysmorphology, and also leads the Manchester Dysmorphology Conference, which will be in November this year. And alongside doing all those things, has led an incredibly vibrant and interesting clinical research program. And today we're delighted for him to talk to us about some of the work that he's been doing in this really very interesting area, which is around chromatinopathies and how episignatures can be valuable in understanding that group of conditions. So Sid, thank you for joining us today, and it's all yours.
Thank you very much, Bill, for the kind introduction. I'm just going to share my screen now. Is it working?
It is. That's perfect. Thank you, Sid.
Great. Thank you. Thank you so much, Bill, for that very kind introduction. The topic for the webinar today for me is from chromatinopathies to episignatures. So these two topics are quite vast, and over the next 40 minutes or 45 minutes or so, my aim is going to be to really introduce these two concepts and topics and give you some details, and especially for those individuals who are somewhat familiar with the field. So it's really more educationally oriented than a research-oriented presentation. So really the plan is in two halves. The first half is about chromatinopathies, and second half will be about DNA methylation signatures. So let's start with chromatinopathies straight away. So as Bill said, in my group, we are mostly interested in neurodevelopment and congenital malformation disorders. So we try to discover these disorders, understand their mechanisms, and hopefully identify treatments and run some clinical trials for these disorders. And broadly speaking, we are interested in chromatin disorders, now splicing disorders as well over the last couple of years or so. And then we're also interested in using multiomics for improving diagnosis of these disorders, and that includes DNA methylation as well. So I really want to start with as to why I got interested in this field. So when I was doing my PhD with Professor Bill Newman, who just gave that very kind introduction, Professor Di Donai, who is a very famous, another clinical geneticist from our department, she had set up a project for Kabuki syndrome. And at that time, the gene for Kabuki syndrome was not known. And by the time I was actually doing my PhD, and it was an incredibly kind of Bill to allow me to work on the side on this project as well, which was quite different from my PhD project. And at that time, as I said, the gene was not known. So we were putting a cohort of patients together for a gene finding project, as you tended to do in those years. So this was about 2010. But around about the same time, Jay Shendure and Michael Bamshad in Seattle, they used exome sequencing, and it was one of the first conditions for which gene was identified using exome sequencing. And at that time, the gene was called MLL2, and I'll come on to the nomenclature of these genes later on, but now it's called KDM6A. So because I was putting this cohort together along with Dai, I thought, okay, now the gene is out, so let's try to understand a little bit more about it. And when I started finding out more about the gene, I saw that it was involved in chromatin architecture, remodeling, etc. So really, that's what I thought. Okay, this seems to be a really interesting molecular mechanism, biological mechanism, disease mechanism, and there are perhaps other genes involved in chromatin remodeling as well. So they must be important for developmental disorders as well. So I started to learn a little bit more about chromatin. So that's the first question that I'm going to address in my next slide now, as to what is chromatin. And again, I'm sure that there are lots of viewers in this webinar who are very familiar with these concepts, but hopefully for those who are less familiar, it'll help to lay the ground for the subsequent slides and subsequent topics in this session. So when you see DNA, which is the usual double-stranded image that we have, and then on this side, you can see the chromosome as a cartoon of a chromosome, rather, as it might look under a microscope. But between these two forms, there are a number of different secondary, tertiary forms that our DNA assumes. And at the basis of it is the formation of nucleosomes, which is this combination of an octamer of histones, H2A, H3, H2B, and H4, and 2 copies of each of these histones around which our DNA is wrapped. And from each of those histones, a tail of amino acids sticks out, which are called histone tails. So just putting all of that into a bit more functional context, here is a slide which I'm grateful for my friend and colleague David Genevieve from Montpellier for lending me this beautiful slide, which really tells you how all of this is relevant in gene regulation and how this is coordinated and how complex the dynamic architecture for this is. So again, this is the nucleosome here with all the tails sticking out. And then these tails have amino acids that can be modified post-translationally, and those are called histone modifications. Similarly, the DNA can be modified. So those are DNA modifications, and especially around CPG islands. And I'll talk a little bit more about it when we come on to our DNA methylation section of the talk. These nucleosomes can slide along the DNA to expose DNA, which might become available to molecular machinery for transcription, etc. And so there are molecular complexes which allow for sliding of nucleosomes, and then changes, the post-translational modifications that I mentioned. There are readers which read these modifications and affect the downstream consequences of those modifications. And all of this is in the context of how the chromatin loops, how it forms territories, how the chromosome territories are formed, and all the intermediates in between that. So this talk is less about the chromosome boundaries and topology associated domains, etc. It's more about the top part that we're discussing, or that's being shown in this particular diagram. So As I said, that this is quite complex. So the next question is how many genes encode the components of these chromatin machinery? And really, like in all of biology, the answer is perhaps depends on what you look at, who is looking at it, and how you're asking that question. And I'd really thank Evgeny Martirosyan, who is a very talented PhD student in my group, who's been looking at gene variations in chromatin machinery. And so this is a figure from his thesis that is currently writing and the paper that he's currently writing. So this is unpublished data. But what Evgeny did was using a number of different approaches, he's questioned the gene ontologies and expression patterns, etc., to identify genes which can be estimated to be associated with components that are important for chromatin machinery. And when you do that, there's almost 1,000 genes which can be confidently associated with chromatin. So it's a huge number. And then there are a number of different subgroups within this number. And this is a UMAP plot of different genes which are in the chromatin machinery and how they can be divided into different subgroups. So there might be some more familiar. For example, lots of you might have come across the methyltransferase and their interactors. So a bit of histone modification that I was just talking about and so on. So, this is just to illustrate that there are a large number of genes which are important for regulating our chromatin. So if there are a large number of genes, then perhaps if there are mutations in these genes, then there must be disorders associated with those mutations. And I've already talked about Kabuki syndrome in one of my initial slides. But what are the kind of disorders? So are we thinking about developmental disorders, malformations, cardiomyopathies, cancer, etc., etc.? So is there any specific type of disorders which are more relevant for chromatin genes? And again, thanks to Evgeny's unpublished work, we've looked at the enrichment of disorder types within individuals who have mutations or variants, deleterious variants within chromatin genes. And what we find is that there's a widespread, and you really find all types of disorders. But if you adjust for genomic footprint of genes that encode chromatin machinery, it's really the neurodevelopmental disorders that stand out. So in this particular plot, this is plotting an odds ratio for mutation, for a deleterious mutation in chromatin gene versus non-chromatin genes. So you can really see that it's about 2 or 3 times more frequent overall than for non-chromatin genes. And actually now we know of approximately of 100 chromatinopathies, so disorders which are caused by mutations in genes that encode components of chromatin regulation. So almost 100 of them are known, and some of these are relatively frequent, such that collectively at least 1 in 500, and that's really a conservative estimate, I would say, 1 in 500 individuals are thought to be affected with chromatinopathies. So that's really, in total across the world, that's hundreds of thousands of individuals, and therefore I would argue that chromatinopathies are an important cause for rare diseases and therefore deserve to be studied in a lot more detail, and we just don't know so much about these disorders. Okay, now because there are 100 of these disorders, as someone like me, I would really like to understand that how can we group them together rather than studying all 100 of them or more than 100 of them separately. So I'm going to use David Genevieve's slide again to be able to show you how at least I think about them. And just as a writer, it's not going to be a comprehensive description of all chromatinopathies, which is of course not possible to do in the limited time that we have. Neither it will be a description of all subgroups of chromatinopathies either, but hopefully it gives you a framework to think, to understand, and to learn more about these diseases going forward. So as I've already talked about, explained this figure, let me gray out the bits that we're not talking about in this section. So we'll start with disorders of histone modifications. So histones, as I said, talked about post-translational modification of histones can take place in a number of different ways. So there is histone acetylation, histone methylation. Methylation can be lysine. It can be methylation of the arginine residues. There is histone phosphorylation. There is histone ubiquitination or simulation. So there are a variety of different kinds of covalent histone modifications. And these histone modifications are dynamic. But what do I mean by that? So once there is a histone, there are enzymes that can add these modifications. So these are sometimes called writers. There are enzymes that can remove these modifications. So these are called as editors. And then there are enzymes that read these modifications as well. But these modifications can change depending on the cell state and age and so many different factors in response to what's happening to the cell, in response to the environmental stimuli, etc., and so on. So these histone modifications are not set in stone throughout the lifespan of a cell or of an organism. Another important property to know about is that we have a really reasonably good idea for many of these histone modifications as to what their downstream consequences might be. So, for example, this is a summary cartoon of histone lysine methylation modifications. There are so many different modifications. I'm just really in this cartoon talking about this part of histone lysine methylation. So lysine in histone 3 tail. So you might hear people talking about H3K4 trimethylation. What does it mean? It means that the lysine at position 4, which is in the tail of histone 3, has 3 methyl groups, so trimethylation. And we know that H3K4 trimethylation tends to be a marker for promoter activation. Similarly, there might be other modifications like H3K27 trimethylation is a marker for gene repression. So you can see that some modifications are associated with gene activation. Other markers are associated with gene repression. So this is sometimes referred to as the histone code, and we'll talk a little bit more about it in a couple of subsequent slides as well. So the next thing I just want to highlight is how do we name these enzymes? So I mentioned about Kabuki syndrome gene. At that time, it was called MLL2, which you can see in this slide here. So about maybe 15, 17, 18 years ago, a nomenclature was suggested for all the histone modifications. So here, I've just taken a snippet. And again, all the slides have references at the bottom, and you're very welcome to dig deeper into those papers if there was a particular topic that you found of more interest. But a few years ago, and some of these nomenclatures still endure. So for example, .1L, we probably still mostly use .1L, EZH2, we still use EZH2. But for many of the other enzymes or genes, a modern nomenclature is sometimes preferred, and it tends to have to some extent a logic which we follow. So again, using MLL2 or MLL4 as examples. So as you might have, if you're really keen, notice that the gene was called MLL2 in the paper. However, the gene is called MLL4 and KMT2D. So one of the reasons why this nomenclature was important, because sometimes there was a bit of confusion as to which gene is called what in which organism. So therefore, this is the reason for that standardization. But if an enzyme is involved in H3K4 modification, then we call it as part of KMT2 group. And then within that, there are different isoforms, so KMT2A, B, C, D, E, and F. If an enzyme is involved in H3K9 methylation, then that forms part of KMT1 group and so on. So that's the logic. So when you're seeing KMT2D, that means that it is an H3K4 monomethylation enzyme. And similarly, there are demethylases. So KDM is K is for lysine, DM is for demethylases or demethylase, and there are different subfamilies of KDM which have specific target residues that they demethylate. So having covered the nomenclature, it's again quite clear that even just for one single group of histone modification, there are so many different genes that code for so many different enzymes. So that means that there are likely to be lots of diseases associated with variants in these particular genes. So in this slide, I'm just trying to summarize examples of diseases associated with histone lysine methylation defects. So we've already talked about Kabuki syndrome, and the two genes associated with Kabuki syndrome are KMT2D, mentioned a few times. KDM6A is another histone lysine demethylase, mutations in which sometimes cause Kabuki syndrome. Then even within the same gene, there might be different disorders, as we all know from so many other examples. So there are specific missense variants in KMT2D that do not cause Kabuki syndrome, but cause a branchial arch malformation syndrome, which is now designated as BCAHH in OMIM. And this really is work of Sara Cupertino, who discovered this disorder and is an excellent postdoc working in my group in Manchester. Some of the other quite well-known disorders involved with histone lysine methylation is Kleefstra syndrome or Soto's syndrome. And again, I'm not going to describe the clinical picture of these conditions because that's not really the purpose for this webinar. But you would probably notice that all of these syndromes are typically associated with learning difficulties. Apart from BCAH, we don't really think that learning difficulties are typically associated, but that needs to be learned. But in a vast majority, learning difficulties are a feature. Developmental delay is a feature. Facial dysmorphism is an important distinguishing feature. Internal organ malformations can be a feature, and growth abnormalities can also be a feature. So, for example, in Kabuki syndrome, there is slowing of growth, and in Soto syndrome, there is a growth acceleration or overgrowth. Then Bill and I jointly co-supervised Victor Fondes, who's an associate professor now at University of Santiago, but he did his PhD with us in Manchester. And as part of his work, he identified mutations in a number of histone lysine genes as cause for neurodevelopmental disorders, which include genes such as KMT2C, which is now called Kleefstra syndrome 2, or KMT5B, which is in some cases can give overgrowth as well, but is also a neurodevelopmental disorder. KDM5B is quite interesting because most of these conditions tend to be dominant. In fact, they are dominant, but KDM5B is an important exception as it is a recessive condition. And again, there are very few patients and much more needs to be studied and understood about these disorders. But these are not just the disorders which I've got put photographs in. There are lots of other conditions as well, and I've listed them here. I won't have time to go through all of them, but then there are some really quite well-known conditions as well, Weaver syndrome, Wiedemann-Steiner syndrome, and so on. So overall, histone modification defects are an important cause for rare diseases, and this is just one of several modifications. And if we go through, let's say, the histone acetylation, again, we can list out a number of different conditions. So I'm just wanting to emphasize the extreme heterogeneity even this particular subgroups offer. But there are lots of commonalities as well. In the next slide, I'm going to briefly and very briefly really talk about disorders associated with ATP-dependent chromatin remodelers, which are involved in either reading of modifications or sliding of nucleosomes. So the first very famous example would be of Coffin-Siris syndrome, which is caused by mutations in components of SWI/SNF complex, or sometimes they're also called BAFopathies, or because they are also called BAF complex. So there are a number of different genes mutations in which all cause this condition called Coffin-Siris syndrome, which is characterized by learning difficulties, hypertrichosis, and nail hyperplasia as well. And there are, of course, lots of other features as well for Coffin-Siris syndrome. The other group of disorders within this ATP-dependent remodelers and readers is disorders associated with CHD8 and PHF6, rather PHF group of genes. So the most famous example of CHD group of disorders would be CHARD syndrome. And again, I don't necessarily need to talk about the different clinical characteristics of CHARD syndrome. This is just to emphasize that this group of chromatinopathies really encompasses some of the most frequent and well-known rare genetic disorders. And finally, in this half of the presentation, I'll talk very briefly about DNA modifications. And as we all know, DNA can be modified. So cytosine can be modified into 5-methyl cytosine by addition of a methyl group. But there are other modifications as well. So some of them are intermediates like 5-hydroxymethyl cytosine. Some are different modifications as well, which are shown here. Even adenine can be modified into N6-methyl adenine. And just like histone modifications and methylation, DNA methylation is also a dynamic process. And there is a set of enzymes that help to add methyl group. There is molecules that read methylation changes. And then there are a set of enzymes that facilitate demethylation of methylation of DNA as well. And mutations in enzymes which are involved in methylation or demethylation of DNA are again associated with some Mendelian disorders. Here I've shown photographs of some individuals with Tatten-Brown-Raman syndrome, which is caused by mutations in DNMT3A, which is a recognizable rare disorder. Again, I'm not going to talk about all the other disorders here, but this is just to show you that similar disorders associated with this subgroup as well. But I'm going to use DNA modifications as my segue into the next section of this talk, which I promised about DNA methylation signatures. So what are DNA methylation signatures? And before we go into that, I would just like to maybe give a little bit of historical background. So I think I would say, at least for me, the story around this started when Rosanna Wexberg and other experts in Canada, they started studying DNA methylation patterns in individuals with some rare diseases. One of the first rare diseases to be studied in this way was Claas-Jensen syndrome, which is caused by mutations in a histone lysine demethylase, KDM5C, which takes away methyl groups from H3K4. And when they studied the DNA methylation of individuals with this condition, they noticed that there was a recurrent DNA methylation pattern. And a thought even in one of those earlier papers was raised that could these actually be could these be utilized as diagnostic biomarkers perhaps for these conditions? But the question really arises that why should mutation in a histone modification enzyme lead to changes in DNA methylation? And to be honest, we don't really know what exactly that mechanism is, but a potential hypothesis is this, that these chromatin modifications, so modifications of DNA and histones, these are interdependent. So they don't, of course, work independently. They influence each other, and they work in tandem, they work in opposition, and so on. Now, if you wanted to study histone modifications, there are a number of techniques which are widely available and widely used. So histone modifications in chromatin states can be studied using technologies such as chromatin immunoprecipitation sequencing, ChIP-seq, cut and tag, cut and run, ATAC-seq, etc. However, to be really utilize these techniques, you need to deal with cells, and they tend to be slightly more labor intensive and expensive as well. So they're not necessarily the easiest techniques to be used as diagnostic biomarkers, and of course they tend to be quite dynamic as well. However, you can perhaps one would not be wrong in saying that DNA methylation is more amenable in terms of diagnostic laboratory approaches, and that's because you're not necessarily dealing with cells in the main part of the assay. You're dealing with the DNA which is extracted already. There are a number of different technologies that are available for testing methylation status, some of which are enrichment based, so usually they tend to be antibody based. Many are bisulfite conversion based. So bisulfite is a chemical that deaminates cytosine such that unmodified cytosine is converted to uracil, whereas methyl cytosine, so methylated cytosine, does not undergo that conversion. And then you can choose whichever genotyping approach you want to be able to distinguish between methylated and unmethylated cytosine. Then there are certain techniques which are restriction enzyme based as well. And now there are new technologies, and I've just used a image of a Oxford nanopore set of different machines, but there are other companies and technology platforms that are available for testing DNA methylation status. Most of the work that I'm going to talk about now in subsequent slides will be it's been done using methylation array. So these are chips, genotyping chips that you can use to interrogate methylation status for a set number of probes, which are usually tend to be spread across the genome. So how are DNA methylation signatures or episignatures derived? So what is the fundamental? And again, it's not a comprehensive workflow description. All I want to do here is to give a broad overview to develop an understanding as to what actually the word DNA methylation signature or episignature means. So just lifting the bonnet a little bit and looking inside. So in the standard human genome, we think that there are approximately 28 million CPGs, so huge amount. Now, I said that most of the work has been done using EPIC array or sort of DNA methylation array, one of which is Illumina EPIC array. And Illumina EPIC array tends to have about 1 million probes, and there are other arrays with different numbers as well. But just as a standard, if you say 1 million, so you're already seeing that it's probably only accessing about 1 in a million, but 1 in 28 million rather of potential sites, which is still super useful. Now, and most of the work that's been done so far in this particular context, I stress, is done on DNA extracted from peripheral blood, and the source of the DNA is important for obvious reasons. But that's important to remember, and we'll perhaps come back to it towards the end of this presentation. Now, you can have, let's say, a set of controls and a set of individuals with a particular disorder. You can run EPIC array on all the controls and all your patients. And here I've again just used Kabuki syndrome because that was the starting point. So we're just continuing that theme for continuity's sake. But if you test these groups of controls in Kabuki, there might be differences in the level of methylation across a few thousand probes out of all those millions of probes. And of course, it depends on what is the threshold that that one would set. But these analyses will need to take into account age, sex, ethnicity of the individuals, because these are some of the variables that impact the methylation of different points in our genome. Similarly, the cellular composition of the tissue, so in this case, blood, is also important because different cells have different methylation patterns. So, and different individuals have different numbers of lymphocytes or platelets, etc., in their blood. So that needs to be taken into account and adjusted for as well. Then you also need to take into account the number of samples. So we're dealing with rare diseases. So it's not like we're going to usually have access to hundreds or thousands of individuals with a particular condition. So the power of your study will, of course, depend on the number of samples, both controls and diseases. And finally, because we're talking about these monogenic diseases, we also need to think about the type of variants that form the cohort of the disease cohort, especially. And the controls, of course, need to be matched with the diseases patients as far as possible. So that background information about the cohort from which episignatures may have been derived is actually quite important. It's very important technically, but it might be quite important for interpreting the results of these tests as well. And I'll try to show a couple of examples for that later on in my presentation. So once you've identified the differentially methylated points, then out of those, a few dozen to 100 of differentially methylated points are selected for developing episignatures. So it's not like all these thousands of DMPs, in short, the differentially methylated points, form part of episignatures. So there are a number of different packages to do these analyses. So many of you will be familiar with Episign, who have proprietary technology company who offer testing across the world. Then and that comes from Bekim Sadikovich's group in Canada. Epigen Central comes from Rosanna Wexberg's group, and then a few newer approaches have also been developed more recently and are being tested currently, such as Methylation. And the purpose for the reason for selecting a few dozen would be that you want to identify those DMPs which give the highest specificity and sensitivity out of all the other several hundred or even thousands of DMPs that you might have accounted for. How you do it is perhaps a topic of a different workshop altogether. So another thing to think about is the last slide was really, we were talking about a single disease, but of course, in clinical practice, you might not be just looking at a single disease. You might be looking at a cohort of patients with different diseases and so on. So it's also important to take into account and make it possible for concurrent detection of multiple episignatures, and all the approaches that I mentioned previously, Episign, Epigen Central, and Methylation, they take into account that possibility of concurrent detection of multiple episignatures. And again, there are, as I mentioned, there is references at the end of each of the slides. So you're very welcome to dive deeper into what this means and how this is implemented. But just to keep in mind to be able to interpret the results accordingly. So having established as to why and how those DNA methylation or episignatures are derived, I just want to spend some time in talking about the potential advantages of using DNA methylation signatures as diagnostic biomarkers, and especially for variants of uncertain significance. And the reason for that is that the biomarkers in our fields, they tend to be not available for vast majority of genes, or they may be invasive or expensive. They may only be available in research laboratories sometimes, and then often not validated diagnostically. So if you're wanting to do an enzyme test, you need to take a skin biopsy, send it to a lab, to a research lab that is studying that particular gene and so on. So it can be quite time-taking and cumbersome, and sometimes just simply not available. Whereas episignatures tend to be non-invasive, so even just a stored DNA extracted from peripheral blood can be used. There is a single platform for several hundred diseases now, and the list keeps on growing, and they seem to have reasonable specificity and sensitivity as long as one is aware of the background and the limitations which we discussed in the previous slides as well. So in the UK, we've been running a genome-wide DNA methylation profiling service to aid variant interpretation, and we've tested several hundred samples, and this is just a heat map which is showing the location of various or rough location of various individuals that have been tested. So we really have been able to offer this service across the country. And this was started about 3 years ago, 2.5 years ago. And really there are 3 possible results. So the testing criteria is that patient must have a plausibly significant variant of uncertain significance in a gene that is covered by this test. And when we do the test, might get a positive, intermediate, or a negative signature. If you want to learn more about our service, I've put a barcode in there. But even if you just put in a search for in one of the search browsers for this, for our service in Manchester, you'll be able to get to the web page. But yes, we've been testing samples from across the UK and abroad. And this is Sarah Hilton, Molly Godfrey, Lee DeMaine, and Chris Campbell, who are part of our lab scientist team who are delivering this test. So this is just a summary, and this is actually almost more than a year-old slide now. We've tested many more samples. So just for VUS samples via NHS, we see roughly in about 40% of cases, we are able to show the VUS to be likely. We are able to show a positive episignature in those cases, and about 55% we are able to rule out that particular disease, which is actually also quite important. So as I said, there are 3 types of results possible: positive, intermediate, or negative. And I'm just going to give you a few examples of all of those 3 scenarios to emphasize the potential nuances of interpretation of these results. So here's a case that was published in American Medical Genetics by our colleague in Johns Hopkins, Jill Farner. So they published a case for of a child who has mosaic for a de novo nonsense variant in KMT2D, Kabuki gene again, and they found mutation in about 11.2% of sequencing read. Now, if you tested a mosaic patient, you might find an intermediate signature. So I said positive, negative, and intermediate, and I'll talk about positive and negative a bit later on, but just wanted to start with the reasons for why you might see an intermediate signature. And what do I mean by intermediate signature? So there are a number of different metrics, but a visual representation could be in this, what's called an MDS plot. So in this, I'll just orient everyone to this plot. The controls are shown in green. The cases samples are shown in blue. And here you can see the red dot in this particular case. So it probably is not really clustering entirely with the Kabuki samples. It's somewhere in between. So of course, the reason for that is perhaps that the level of the reads with the variant is lower than normal 50%, as you would expect. Then here's another example. So we already talked about Class-Jensen syndrome. It's an X-linked disorder, and what's been shown by Bekim Sadikovich's lab is that that female carriers for KDM5C Class-Jensen syndrome, they tend to have a DNA methylation profile signature, which is somewhat in between controls and cases. And again, not very surprising as to a being a carrier might be a cause for having an intermediate signature. Then this is a study from Rosanna Wexberg's group, and they studied a condition called Alpine syndrome, which is another Kabuki syndrome-like phenotype caused by mutation, caused by one of the heteronuclear ribonuclear protein mutations. And what they showed was that they studied the missense variants in the gene, and they found that patients who had missense variants, their DNA methylation profile was somewhat intermediate between the controls and cases which were made of loss of function variants. And they also showed that some of these patients have perhaps relatively milder phenotype. So these are perhaps hypomorphic variants, and therefore they're giving some of these hypomorphic variants in some of the diseases may give rise to intermediate signature. So that's an important point to remember. If you're seeing intermediate signature, those are some of the causes. There are some other reasons as well, but we probably do not have time to go deeper into this. I would also just caution that when we are interpreting negative signatures, what might be the reason? So here's a paper from Murad Ansari and a number of other individuals studying SMC related Cornelia de Lange syndrome. So most of these individuals have missense mutations, but they found certain patients with stop mutations. And when we tested the episignatures for these individuals, we found they're not clustering with the Cornelia de Lange cohort here. And the reason for that is we think that probably the disease mechanism on missense variants versus loss of function variants might be different. So although these variants almost certainly are pathogenic, they do not show the episignature because these episignatures are not necessarily disease specific. They are disease and variant specific. Sorry, these are not gene specific, but they are rather disease mechanism specific. So again, seeing a negative signature doesn't necessarily mean that the variant is not pathogenic. So knowing what is the disease cohort made of against what you're testing is a really important concept. And then positive signatures are relatively straightforward, I would say, where you see a positive signature and that perhaps is easier to interpret. However, there are certain unusual scenarios which would be useful to be aware of. So for example, it's a case of a 2-year-old boy I saw in my clinic who was very typical for Kabuki syndrome, but he had a deep intronic variant in KMT2D. We were not able to show that it was de novo because one of the parents was not available. So, but the phenotype was very typical. The splice AI scores, etc., for this were reasonably convincing. So we thought that, okay, this might be the answer. This variant might be the cause for this patient's phenotype. But when we tested the DNA methylation signature for Kabuki, the red dot here is the patient. The blues are all the Kabukis, and it fits very nicely. So we thought, yeah, okay, we've got the answer. But we're also setting up RNA studies at that time. So we took a sample from this child and set up some RNA studies. And interestingly, the RNA testing did not show any aberrant splicing. And I don't have a plot for this presentation right now, but you just have to trust me that that's what it showed. So by that time, so initially the Kabuki syndrome signature was just for Kabuki syndrome, and in one of the subsequent expanded versions, we now have from Episign Kabuki syndrome type 1 and type 2 signatures separate. And that showed, so we just rerun the data on the new classifier, and that showed the child was negative for type 1 Kabuki syndrome, which is the KMT2D type of Kabuki syndrome, but was positive for type 2 Kabuki syndrome. So that was really surprising. But because we had done actually targeted testing, and this was several years ago, KDM6A was never tested in this child. So we then did targeted analysis of KDM6A, and we were able to find a mutation in KDM6A, which was much more likely to be pathogenic. So here was a positive signature, which was positive, but the results were slightly unusual and unexpected. But finally, we did end up giving the right diagnosis. And it's important because KDM6A is an X-linked disease, and occasionally mothers can also be carriers for this condition. Here's a wonderful case from Slavica Trachova and Alfredo Brusco's group in Italy, where they've shown a very interesting case where a patient with a mutation in a gene called SMARCB1, which is a baphopathy, which I've mentioned earlier in my chromatin part of the talk. But the signature, we thought that the mutation must be pathogenic, but the signature did not cluster with the baphopathy signature. But instead, it clustered with a different specific subgroup of mutations which affect ARID1A mutations, a very specific mutation in ARID1A affecting the nucleotide number 6200. And then when you look at the, Slavica and Alfredo looked at the molecular complex, they could see that this particular residue in SMARCB1 and in ARID1A, they really interact closely with each other. So it's not just the same gene giving rise to same episignature, but actually testing episignature here tells us that it is really the mechanism is more specific to perhaps than simple loss of or simple alteration of function in SMARCB1. So again, a positive episignature, but not as expected for baphopathy, but you end up really discovering potential very interesting disease mechanisms as well. I noticed that I'm perhaps running out of time, but just wanted to quickly point out some of the work that Camille Dupont, a lab scientist in our group, has been doing. So she's been exploring the utility of DNA methylation profile in unsolved individuals without a variant of uncertain significance. And I would probably want to emphasize using a case here. So I saw a child who we suspected might have a condition called Burson-Feynman-Lehmann syndrome, BFL, but the whole genome sequencing did not identify the mutation. So we ran DNA methylation profiling, saw an episignature consistent with BFL, and that led us to identify a de novo PHF6 deep intronic -1454 mutation in the gene, and then which we were able to prove using RNA sequencing. And sorry for running over this rather quickly, but here's an example how whole genome sequencing, DNA methylation, reanalysis of data, and RNA sequencing all work together to identify cause of a child's medical issues. I'll probably just skip over. I just wanted to also make people aware, for those who are interested. So MRC have funded a group, a node called EpigenRare, Epigenomics of Rare Disorders, to perform more research into the disease mechanisms and finding treatments for chromatinopathies and imprinting disorders. So we are very excited about the work that this group of that we might be able to do. But we've also established, as part of this EpigenRare research group, we established an EpigenRare network, which we want to develop as a community of like-minded academics, healthcare professionals, and representatives from industry and patient support groups to address the many remaining challenges, as I mentioned at the beginning, understanding, diagnosing, and treating of epigenetic rare disorders. So we held our first conference, EpigenRare conference, earlier this year in Manchester, and at the ESHG next year in Gothenburg in June, we'll host a EpigenRare symposium. There's also an Episignatures workshop, which is not part of EpigenRare, but an Episignatures workshop that there was one held in 2025 in Milan, and there'll be another Episignature workshop for those who are interested in this. Please visit that workshop. This was only like 15-20 minutes of that. That workshop would be 1.5 hours. If you were interested in joining the EpiGenRare network, here is a barcode you can scan, and you're very welcome to be part of this growing network, which we have people from across the world. I'll move that barcode to the next slide as well at the top now. But in summary, we've chatted about what are chromatinopathies, what are DNA methylation signatures, what are their current applications. I skipped the future directions and I apologize for that. There are lots and lots of people to thank. I mentioned them by name throughout my presentation, a lot of our funders as well, and all my mentors who have helped me to put this program of work together. But of course, most important, all the patients, families, and contributing clinicians. Thank you very much.
Sid, thank you so much. It's absolutely wonderful, and such a huge amount of work that's gone into that. We've got time for a couple of questions. So can one of the questions that's come through is from Karina, just asked, do infections alter these signatures?
So that's a really excellent question. Environmental factors will alter episignatures, so exposure to drugs and perhaps to infections as well will alter episignatures. So one does need to be aware of what the patient's current status is at that time when the sample was taken. So if you think that the result do not match your expectations, then it's worth spending a bit of time and thinking about how the patient was at the time when the sample was taken. And an important aspect in that is for most of the diagnostic biomarker work that is currently available, you would prefer the sample to be taken not very early on in life in first few months, because again, all the classifiers have been developed mostly using older individuals. So the age of the patient at the time of DNA methylation is important. At the time of DNA collection is important.
Yeah, very, very helpful. And question from F. Manzi, who's asking, is there a database which sets out all these episignatures for different conditions that you can interrogate?
Yeah, so for example, I mentioned EpiGen Central and Methodori. So these are some databases which are publicly available, or at least part of it is publicly available. Yep. There are certain papers, research papers, where the data has been made available publicly, but there is also a lot of data which is proprietary and not available as such. So it's a combination, I would say.
Yeah. And Sebastian Silva asks, does HERC2, HERC2, have a well-characterized episignature?
That's a question I don't know the answer to specifically, but I can look it up and, I guess Sebastian.
Yeah, it's around that. Perfect. Okay. And then the last question that we have is, would you consider cohesinopathies such as Cornelia de Lange as a chromatinopathy?
Absolutely, yes. And again, I did not mention it because of lack of time. But yeah, for example, all the cohesins, they are super important for gene regulation. So they're very much part of chromatin disorders. And another point I just wanted to make in response to that question is, I've kind of implied that DNA methylation signature is only relevant for chromatin disorders, but actually that's not necessarily the case. A majority of these are perhaps chromatin disorders, but there are lots of non-chromatin disorders as well for which DNA methylation signatures are known. So yeah, and again, because I didn't have time for future directions slide, some of that was missed.
Sid, thank you so much. There are some other questions that have come through, but we can pick those up afterwards. It's been a fantastic webinar. We really appreciate it, and it's sort of shown by the large number of questions that have come through. Our next webinar is on the 24th of September, the same time, 4 p.m. Central Eastern European time, and it's Serena Nick-Zainal from the University of Cambridge, who's going to be talking about mutational signatures. So we look forward to joining then. You'll be able to revisit Sid's wonderful webinar on the YouTube channel. And so please take a look if there's anything you wanted to spend a little bit more time concentrating on. Thank you very much for joining us today.
Thank you very much, Bill, for the very kind invitation.
You're welcome.
Bye.